Okay, so again, the terminology, I briefly introduced it already on Tuesday. Physical protein interactions are those that happen in contact between two proteins that go on and off. So the idea is that this is a transient interaction, that this is something between proteins that are not, if you sort of want to misuse that word, born together. So the green protein here, the GP120, and the blue one, the CD4, they have to meet first, and that's the idea about a transient protein interaction. Transient, I'm making such a stress of it because transient could be for the lifetime or for most of the lifetime of the protein. Could be that this complex exists, this particular one does not exist that long. That's a relatively short one, but some complexes exist longer. So these are like machineries that are formed, but they're still transient in the sense that the two proteins first have to come together. Okay, and that's what we're looking at. There is a very different sort of thing that I refer to as association. Often that is called protein interaction. In a chain such as this, A activates B, activates C, activates D. D and A are associated, but not really interacting for me. Okay? So these two are not physically interacting. These two are. Okay. Now, the way you typically measure, so most of the measurements for today, what we talk about is really based on three-dimensional structures. But most of the measurements for protein interactions are done by something that is called yeast-to-hybrid system. So the way you want to study whether X and Y bind, and the way you do it, you put a reporter binding domain and an activation domain. So both to X you link a reporter, and to Y you link a reporter. Reporter is simply, this is an element that you somehow can control. You make, in the genetics of whatever algorithm, yeast to hybrid was started in yeast. So in yeast you can control what you put on top of X. Okay? Now, whenever X and Y interact, then ultimately these two being close to each other will emit a signal, something that you can actually see. That's the idea. And you immediately see that there are all kinds of problems. So the signal thing, the binding domain here, is not just a small thing. There's another entire protein, right? So it is not that difficult to assume that this may actually have an influence on whether these two bind. And it could be that, in fact, the way you put B on there is sort of interfering with the interaction. By going into the binding site there, so there are all kinds of problems. But still, in the high throughput sense, this is the best kind of system that is called yeast-to-hybrid system that we know. And there are many others, and I'm not going to go into this. So let's go back again to this issue of homology-based inference. In homology-based inference for protein interaction prediction, essentially we have these four proteins now. So for all the other cases, such as enzymatic activity or localization, it was very simple. We said that the sequence similarity between Q and X is larger than the threshold T, and if for X I have an annotation about function, then I can infer that Q also has the same annotation. Now here the story is slightly more involved, because now we're not only having two. So here we have the case that between proteins A and B, I experimentally know that they bind. This is actually the X in that story here. And then I have another pair of proteins, A prime and B prime. So they symbolize the Q. Those are the query proteins. And for those, in fact, I want to know whether they bind or not. And I want to know that, again, depending on the threshold. So we do the same thing as before. We create a data set that is somehow redundancy reduced, and we look at what sequence level threshold we can see what kind of conservation. Now in this particular case, this is what you get, and this is sort of the HSSP threshold. Remember, I showed you this curve that separates out proteins that have a similar structure from those where we cannot say that it has similar structure. Number of residues aligned, percentage sequence identity. The zero value here is this blue curve. That is +5, +10, and so forth, and that is -5, -10, and so forth. And essentially only plus values are printed here, are shown here. And you see, yes, homology-based inference goes up. So for whatever is within that error, the slide this plot is 12 years old now, so there was not that much data. There was sort of an increase but it was not very rapid. Let's not hang too much. Well, let's just stay on what this axis means. This is accuracy. 2%. So where so far we talked about 90%, now we have 2. But here the story gets more complicated. Because for the data set that we had, we said that if the protein is not known to us, we call it not known. But we also knew at the same time that most interactions were not known. So there's a high error margin in the way we estimate that number. So all that really is relevant at this point is, does it go up or not? And we see sort of there's something going down, but the error is so high, as you see, we can see that this is smaller. But even here, the error bars completely overlap for all of these points. Now this brings up this discussion of homology. I briefly alluded to that already. The original way of using homology is in organs or entire organisms, where you see that there is an there's an organ, in this particular case here, the hand and arm, that looks somehow similar in different organisms. There's a homology, although very few of you have most likely seen whales walking around. It actually looks very similar, arguably, right? So there is an organ that looks very, very similar in human and whale, and that ultimately is this idea of homology. They are related, they are evolutionarily related. On the level of proteins, we discussed this is slightly more complicated. In this particular image, we would have species A here that has, for the sake of the argument here, for the slide, has 3 different proteins, and then there is a species separate into B1 and B2, and B1 and B2 essentially, since that's the ancestor, they have the same set of genes. Okay, and then things are duplicated here, and upon duplication, maybe so this domain here is duplicated and slightly changed. When we go to the next species C here. So we have 3 generations in this image here, 1, 2, 3. And at one of these steps here, there was a duplication of a gene. So now, when all you see from today is essentially these 2 images here. And we try to infer whether there's a relation here, because we try to see whether this relation between that and that domain goes back to a common ancestor. That's somehow the idea of homology. Well, you could immediately sort of kill this entire idea if you had jumping genes, right? So if you imagined that genes would sort of sit there and jump out of the organism and to the next one, right? Luckily, this is nonsense, right? Is it? This is being biology, right? No, it's not nonsense. Jumping genes exist, and in fact, the first time they were discovered was in corn. And that is not the quote I have, unfortunately. I should have had that quote in here. So you see that some corns have these different colors, and ultimately this corn cobs, if you look at corn cobs, here typically they're all yellow. So here I don't see that often, but in the US, if you go to a supermarket, you see them multicolored. You see them maybe on the Day of the Dead in Mexico, when you see movies of Mexico, and so anyway, those are results of jumping genes. Here is an example of a sea slug that incorporates not only genes but entire organisms, or entire parts of other genes. In the world of bacteria, there's a lot of exchange of genes, but there's really also very strongly this situation of jumping genes. Single genes that jump out into the other organism. Part of that is an actual active defense mechanism against the other one. Now, this brings up the following problem. When I said that essentially we know that A and B bind, and I want to infer that A prime and B prime bind. Now, actually, the question becomes, well, say there's another A2 prime and another B2 prime. The A prime and B prime are in the same organism as A and B, and A2 prime and A B2 prime are in a different organism. So now the question becomes, not only can I do homology-based inference for protein interactions, but which one would actually work better? So this is the kind of thing that I would typically not expect you to sort of get an idea what you believe will work better. But let me try it nevertheless. So let me put it differently. If you make a mistake in your answer, this is entirely my mistake. If you get it right, then you got some biological intuition that you most likely came to the course with. What do you expect? So again, my question is, we're going to look at the same level x here. So the difference between A and A prime is the same as between A and A2 prime. By the way, what I plot on the other graph, what I showed there, was the minimum. So you could look at the average. So there are obviously two distances here. While in this simple example, you have only one sequence similarity that matters. Here I have the one between A and A prime, and the one between B prime and B. And now you could sort of average these two. You could look at the min, let the max. What I showed you is the min. Again, let's assume we look at examples where A and A prime and A2 prime are essentially the same. And I'm asking, is this homology inference between A within the organism going to work better than between organisms? Any guess? Any guess? Yeah?
I think they're the same. I don't know. I have the intuition.
Because you don't see any reason why they would be different. Okay? They're not going to be different. There's a biological expectation and there's an observation. And these two things are opposite to each other. But none of them shows the same. You have to make a... So either you fall on the side of what... If you take a different side than the one, they are the same. Any other answer either is the one that everybody expected or the one I'm going to show you. One of those two. Any argument? Okay, let's begin with the data in this particular case. So this is warm versus not warm. So that's the case here. What I showed you before is the one between A and A2 prime. Let's just put the other one on top of it. That's inside that organism. Wherever you are, there are big error bars, but it seems to be very clear that they are different. So why is that surprising? Most people would have expected the story to be exactly the other way around, because when you sort of have the speciation event between these two, then you copy the entire genome, and essentially this other species does the same thing. They need the same proteins and most likely the same interactions. While if you have an additional copy inside of your organism, you're essentially free to evolve some new function, because you anyway have the A and B. Why need another copy? Well, if you need the same thing twice, okay, it would give you some additional support, but that is the entire sort of image of biology, that this is the way we can evolve new functions, that we have a duplicated version, and the duplicated version is no longer bound by having to do one particular thing that already is done by A. Because essentially you duplicate something and you're now free to evolve, because A does it, whatever you need to be done today, A does anyway. And if this one here changes slightly, you have the opportunity that it does something slightly new. Okay? Now something slightly new is essentially implying that this function here is less likely to be maintained, right? So this is very, very surprising. So we published this 12 years ago, and why this is the case, we actually still don't know. Now people meanwhile have discovered, so people have discovered, or have published this finding itself again, have discovered a similar finding for other aspects of function, enzymatic activity, for instance, aspects of Go annotation, but why we don't quite really understand. But let's get back to the issue of predicting protein-protein interactions. So ultimately what I want to do is I want to take one protein here and another protein and somehow show where these two proteins are binding. And the first way in which I did that is I simply put this into, about 20 years ago, I put this into a neural network. And simply, so that had, in this particular case, what you see here is 3 consecutive residues from one protein and 3 consecutive residues from the other protein, and then you just slide through both proteins along each other, right? And you tried all possible scenarios, and that is the first way in which I tried. And that was a complete failure. It absolutely didn't work. I claim it absolutely doesn't work. Some people have published something like that, and I still believe it doesn't work. And my argument is ultimately that you have so many false positives, because when I take in the computer these two proteins and I slide them by each other, I have things that will never happen in nature, because these proteins are folded. The only thing they will see is somehow their surfaces. Okay, they sort of are dynamic objects, so they open up a little bit. The surface changes, and that is crucial for binding. I know that. But it's still true that a lot in that protein will not see the other protein. Okay? That's one reality. Another reality is, well, what else could be the reason why two proteins don't bind? So they have something that totally fits in a computer, looks like they would bind, and in reality you don't observe that.
They are not living in the same environment.
In the most extreme case, we talked about localization. Exactly, ultimately for that reason, one is nuclear and one is secreted. They will never meet each other. So if you put them in a tube, they would bind possibly, but in vivo, in the cell, they will not bind. Okay, that's another aspect. So in this experiment, I completely ignore these two realities. So the first thing that we should actually do to somehow acknowledge these realities is, if I know that the two bind by surface, then I should sort of predict surface. Let's assume I have a surface where sort of half of the protein is inside and half of the protein is outside, and the half that I have outside is sort of the one that is more likely binding. Then you see I cut the false positives by a factor of 4. By not comparing two entire proteins, but only two of their halves on the surface, essentially I have only 1/4 of the... So this simple decision, because it's squared between the two proteins, right? So with a simple decision like that, I can immensely cut the false positives. Okay, in saying that, I will also lose some. But I will throw out so many more false positives that I hope that the signal that I get in the end of the day will be beneficial. And in fact, for that I have to sort of first predict solvent accessibility. Last semester I briefly, in passing, did I discuss about solvent accessibility? Maybe not even. So sometimes in the structure, not sometimes, usually in the structure prediction lecture, I talk about solvent accessibility prediction. But in the structure prediction lecture, so if you have the goal to predict structures, what you want to predict best of your protein is the inside. Okay, so if you want to predict, if you want to develop a machine learning device that predicts surface or residues that are inside and outside, this method is always, in the interest of predicting structure, is always focused on getting it best inside. Here is the other way around. Here I really need a method that is very good on the surface. So in fact, we had to redevelop a method that is sort of focused on surface. We did that. Then the next target was, this gives me the surface residues. What if I could, instead of only looking at the surface residues, could actually predict which residues would most likely bind on the surface? So I could sort of predict the binding sites, and that would sort of cut my false positives even further. And this is what I will talk about today. But let me first continue on this story here. The next so we developed that at some point, and then came this idea of localization. We talked about it already. Proteins have to be in a similar compartment to bind with each other. Here we have the work that we talked about, log tree 2 and 3, that essentially has 18 different classes. So in 18 different classes, just assume if you only predicted 5 localizations, you already reduce the false positives by a factor of 25, because it's a square thing again. For 18, the number is 18 squared, right? It's not quite true, because these classes are not all equally populated. So some of these 18 classes are really small, but it clearly reduces the false positives. So the next idea was to predict protein substrate interfaces, and this is something that we did in the past, and we are still actually doing. It's still active work. We had to work on better domain and better alignment methods. And then ultimately, I showed these slides for many, many years in my talks. Not in my lectures, in scientific talks I gave. And then I always said, and now all we need to do, so we have developed all of these tools. And just to give you an idea what goes into this slide. So this is over $10 billion spent over 10 years. Over 15 people worked on this. And now we have it all together, and I always showed this slide, and then I said, and now we're going to put it together and we're going to predict protein-protein interactions. What happened over the years, the slide didn't change. So this already gives away that something went wrong. The second thing that went wrong is that the phase changed. So I always had some person who's going to nail it and complete it. And every 3 years, the phase was a different one on my slides. So there were many, many things that were challenging. Ultimately, Tobias here is the one who actually nailed it. And this is what I'm going to talk about. I'm not entirely sure whether we get to it on Tuesday or next year. Let's begin with the data collection. And let's now talk first about this issue that I want to predict which residue binds to another protein without knowing the binding partner, without knowing the structure. Again, the ones we are going to extract are these here. We're going to extract those that are close to each other. There's something missing here, or something wrong in this slide. So we're looking at things that are less than 6.5 angstrom. So the closest atom is 6.5 angstrom, that we call connections, and then we are going to collect all of these connections, and we're going to ask, do they differ between residues between two different proteins here, or between similar proteins, or within a protein? So this is essentially a homodimer. So we're going to, in the first step here, distinguish 3 different classes. On my next slide, I believe it's 3 classes. Yes, it's essentially inside a protein, between 2 transiently binding proteins, so proteins that bind sometimes, and then a binding between essentially 2 subunits of the same thing, the same protein. Okay, and what these are the 20 amino acids, somehow sorted by biophysical features. This is a surprise, a frequency, a log odds ratio, which means the surprise. How surprising is it to see that amount of R residue in that particular interface, in one of these 3 interfaces, given how many R's you have in a protein? Okay, that's the background frequency. So above 0 means you have more of them than expected, below less than expected. Blue is again chain, so it's essentially homodimers. Green is the internal stuff. Red is the transient protein-protein interaction. And again, so if these would be the same, green, if all 3 would be the same, then I could, without any label, look at the entire PDB and use the entire PDB to develop protein interaction prediction methods. Now, what I'd like to do is that everybody, this usually is the moment when you say, fine, publish. He said some doubt, ultimately because he did not completely trust statistics. What could you do? What could you try to do if you sort of don't trust that very well? I had a very simple idea.
Try yourself.
Try yourself? What do you mean?
Like you don't trust that.
Well, so reprogram it. Fair enough. This is a computer science answer. Absolutely. I take your point. Say that give the same result. Yeah?
Try the same test on subsets and see if the difference is similar.
Exactly. And that's what he did, but he did it slightly differently. He did not quite use subsets. Instead, he completely ignored the classification into different interfaces and said, let's just look, what's the difference between proteins that have the first letter being A or B and the first letter being C or D? Oh, and let's just look at these as different sets and see how significantly different are they. Or let's just look at sequences from people where one author published it, or 2. And then we went to the next step, random subsets. And guess what? Essentially for all of them. Certainly. So some of them random was much lower than that. But it was still smaller than 10 minus 100. So everybody typically is absolutely content with 10 minus 5. And we were beyond that. Interestingly, by the way, this here is nonsense, this is nonsense, this is nonsense. But then actually we saw that when the number of authors was larger than 10, there was biology behind it, because then the protein was longer. And actually then, so longer proteins were sequenced by larger groups, and there was really significantly something different. But most of these were nonsense, okay? So now the question is, why is the chi-square test going wrong? One possible reason is our data is too large. But another problem in the chi-square test that I did not show here is it assumes that the distribution that you have is unrelated. These two distributions are unrelated statistically. So what we're talking about here is two different subsets of protein binding sites, and they are evolutionarily related. So we cannot clearly claim this unrelated statement. Now, if that really would be the reason to explain why this goes wrong here, we would have a problem. It would essentially imply that in the entire field of biology we cannot apply statistics. Obviously, we don't believe that. But we also don't know what went wrong here. So we reverted to a different idea. And the idea is very simple. It's the following. So we have three different data sets. In our case, this is internal homodimers and external interactions. And these different balls in here are essentially the 20 amino acids. So we have to, in each pot here, you have 20 different colors of balls. All right. Now what you're going to do is you're going to pick out some subset from this, the same subset, I mean the same sized subset. Say, just for the sake of the argument, 1,000 from here, 1,000 from here, and 1,000 from here. Then you're going to pick another 1,000 from the first, and you're going to compare these thousands, this to that, that to that, and that to that. And you're going to ask the set of 1,000 by some Jensen-Shannon information. Are these thousands most similar to that, most similar to that, or most similar to that? And you're going to do that many times. In fact, again, I'm going to do that 1,000 times.
What does JS?
Jensen-Shannon information. So it's essentially P log P. Something like that. I mean, you simply have to compare this. It's a multivariate state essentially that you have to compare. You can do it in different ways. Jensen-Shannon is one way. Okay, is the process clear? Now on the next slide, what I'm going to show is the results from this. But I'm going to, instead of having 3 states, as I showed you in the previous slide, I'm going to have that for 6 states. Those are the 6 states. Let's begin to explain what these 6 states are. This is the thing that we had before. But again, the numbers here are, I take 1,000 from internal and I find most likely internal. In 909 of the cases I find internal. In 65 of the cases I find domain-domain. Okay, domain-domain, what does that mean? So this is really inside of a protein. Domain-domain means I have 2 domains in a protein and they interact with each other, which is still inside the protein, but it's sort of between 2 distinguishable subunits inside the protein, right? These here are both homodimers. So these are interactions between, you take the protein, make another copy of the same protein, and they work together. And there are 2 versions of this. This is what we dubbed, or it comes from Janet Thornton, the idea, we call that obligomer and oligomer. Obligomer are things simply where we see this, the same contact between the same. Exactly. So this is the original protein, and there's a second copy of this original protein, and these 2 bind. Okay? And I never see this protein bind anything else, only to itself. Then I call it obligomer. It's obligatory for that protein to bind itself because it doesn't find another binding site, binding partner, whatever the reason. The oligomer are simply the same thing here, but in that oligomer thing, this also binds something else. Okay? Now here, I call it heteroobligomer. Now this is in fact the situation where I have a protein that has 2 different parts, 2 different chains. That is more than domain. They are really more separated than domains are separated. But they are still part of the same gene. They're co-expressed, they coexist. They are different in terms of the sequence. and they bind. I call them obligomer because they cannot escape each other. They are connected. Okay? In an evolutionary sense, they're coming together into existence, so they're co-expressed, as biologists call that. And they, as long as nobody cuts the chain, they are always together, right? In that sense, they are obligatorily together, and they actually bind together. While the last point here, the heterooligomer, so hetero means two different sequences, right? The heterooligomer, these are two different proteins that bind transiently. Okay, now that this complication is out of the way, what you clearly see is that the diagonals are the highest. So in a statistical sense, you can absolutely distinguish. You find the class you belong to. So statistically significantly, they clearly differ. By the way, if I did that now for random, the entire matrix is pretty much equally populated, and that's true for all the other ways in which we split. So splitting the subsets still, getting back to your original idea, Tobias, so looking at subsets, the matrix still survived. At some point the data became too small, and then the matrix no longer survived. But until that point, actually the matrix survived. Now, let's look at the confusion points that I have, meaning, so yes, the diagonal elements are always the highest, but the highest level of confusion is this. So this is, of all the diagonal elements here, the 812 and the 811 are the lowest. And let's just see what that is confused most often with. So clearly there's a confusion here to internal contacts, and that makes sense. So domain, internal is both internal. But here is a confusion to something that is not quite internal. But remember, heteroobligomers are things from the same protein entity. So they are together in a biological sense. They don't have to meet. They're co-expressed. So in some sense, these two things are not that different. Whether you have a domain inside that you can recognize, or you have two more different fragments. So this confusion, biologically at least, makes sense. Remarkably, they are really different. In some sense, the confusion here is high here. And the other confusion they have is with homo-obligomers. So with things that form a homodimer and are always seen to bind with each other. So this confusion we cannot explain. There's nothing to be said about it. But clearly what we see, another observation that was remarkable to us is that homodimers stand out the most. So that is the most unique way of forming contacts. But what this now means is they differ, and if they differ, it means it's different physics, and it means actually, back to what Francesco said before, or implicitly what Francesco says, if they differ, it means I can actually predict. So this one here is just statistically showing that there's a difference, right? Doesn't mean I can predict, because I take out a subset of 1,000, and from the subset of 1,000 I can predict. But that is not meaningful. What is meaningful is in my particular protein, can I predict them? Okay? Okay, how do we do that? And again, so we're trying to first of all predict external protein-protein interactions. For that we gather the PDB and we extract the interactions in the same sense that I said before, less than 6.5 angstrom, parse out some heavy atoms, and there are some additional complications. Here's one, let me cut out through that one too. There is this problem with choosing a threshold that I talked about briefly, and I'm going to cut through that one too here. And let's get back to the machine learning. So the real question is, how do you choose the input features? So now I want to predict, I want to have a method that uses sequences input and predicts for a particular residue. Say I take out a window of 5 consecutive residues. So in this lecture we still, so far we haven't had the idea of a window yet, the function prediction. So what I'm going to do, again, I have my entire protein here. And from the protein I take 5 residues out, these 5, and I want to predict whether the central one in these 5 is binding in an external interaction or not. Okay? So essentially I have a thing that has 5 times some input features, input, and 2 output features. It binds externally or not. We could do that in one, but again, typically two in our experience do better. How do we find out what input features we take? Well, clearly I'm going to use the sequence in some sense. So one way to code the sequence, the simplest way to code the sequence is you use 20 amino acids. So that means I have 5 central residues times 20. Now 20 is not enough because you need a spacer. Just briefly for the ones who are new, I said I want to predict this for the central. If the central is the first residue, then I have 2 above here that are not amino acids. Now I could do that by sort of making them all 0, saying this is not an amino acid, but actually it turns out that it doesn't work that well. What works better is if I added another unit and call it the spacer unit. That is 1 for something that is not an amino acid. Okay? Those of you who have been last semester's lecture, that is all trivial. But now my question to you on this slide here is, what else do I want to put into the system? What kind of input information? I'm sorry. So what you see is, in many cases here you have in fact 9 and 9. In more you have at least sort of 5 in a stretch. They are not consecutive. In a stretch of 9 consecutive, you have at least 5, and that's somehow 5 or 6. That's where it's peaking, right? And the story is slightly different. The distribution in yellow when you have a more strict threshold, and so we use that to refine the prediction. And this is ultimately what we get here. Then we put the elephant into it, and we get the dark blue. And the dark blue, essentially what I had before, I blew up the scale here. Now this is the real scale, the full scale. Sequence only has some signal, but not much coverage. The complete dark blue here has a large coverage. And that brings up the question of hotspots. Now hotspots, essentially the idea here is we have residues that are essential for protein-protein interactions, and they are called hotspots. So essential means, for instance, I knock out one of these hotspots and the binding is changing a lot. Okay? So the problem is that some people argue hotspots don't exist. What you see is that you have a logarithmic function. So when you plot here how much, this is a log plot now, when you plot one single residue contribute, no wait a minute, I have to do the other way around. No, I can do it this way. So the residue that contributes the most to the binding energy may carry, maybe this point here is 30% of the binding energy or something like that. The second one maybe 15%, and so forth and so on. So one way of arguing now is we have overall, this curve here goes to 20 until the entire binding is done. So 1 in the 20 carries 1/3 of all the binding energy. So this is why I call that a hotspot. And other people say, well, it's just a function. Some contribute more than others. There's no point in calling them hotspots. Hotspots gives this illusion that they stand out. But this is essentially a chemical sum, and everybody contributes something, and there is no point in putting a cutoff. There's no point in saying this is more relevant, this is more relevant. This is just a straight line, right? So biochemically, then the argument continues. Hotspots are not significant. They're not standing out biologically, right? And that has been an argument that has been debated in the literature of experimentalists for a long time. It's relevant because pharmaceutical companies always target the hotspots. Typically, they want to interrupt bad protein interaction that has to do with an intruder or has to do with something that goes wrong in the body. And you always do that best by attacking the hotspot, the most important one. If you could sort of have a drug that goes exactly into protein residue 1 and 2, you would sort of possibly derail the entire interaction by just knocking out 50% of the energy of binding. Or at least you would reduce the probability of this problem that occurs by 50%, right? By just targeting 2 residues. But again, now comes a different question. So I did show you this plot here. Recall precision. The question is there anything special about these residues up there? So there are some residues here, and okay, there's noise, or somehow if I had a larger data set, most likely what I would see is some sort of function such as this one. So most likely what I see here, the sudden dip, is something that relates to a problem in the smallness of the data set. Still the question is there anything special about the residues up there? What if I only looked at those? And when Jan and I did that, there was somebody in the other room he had lunch with all the time, and they happened to have experimental data on hotspots for the same complex of proteins that I showed you before. In fact, because that group did that complex, CD4 and GP120, and the red ones here are observed to be most important for the interaction. Okay? And that's the only set he ever looked at that point, because he was asked this question just briefly before his defense. And Wayne Henriksen was in the committee. And here's his prediction. So that prediction only uses sequence. So what I'm showing here is just put the prediction onto a 3-dimensional structure, but that information is not used. Okay? What you see is the prediction is purple. You see that purple and red, the observation here, are very similar. You see regions where you underpredict, so you miss this one here, right? There are regions where you overpredict. This one in the back here is wrong. This one is wrong. But all somehow, so you pick up the major theme there, and the mistakes you make are sort of surrounding this major theme, right? So this was totally remarkable, and ultimately this brings us to the point, if we can predict these hotspots, the method never used the idea of hotspots. If you can predict hotspots in a machinery that never learned to predict hotspots, so a machine learning device only looked at differences of protein interaction sites, and it had figured out that some stand out biologically, they're more reliable, they're more consistent. Ultimately on the level of machine learning is only an issue of consistency, right? But this consistency, if it is in the data, tells you that there's something in the biology, right? In some sense, so we could predict hotspots without training on hotspots. Or we just picked as in the rock-like curve here, the top 10% or something like that. We could have picked the top 20, top 5. All of these gave essentially amazingly good answers. So again, depending on what fraction you pick, you get different accuracy, different precision recall balance again. But all of those showed that essentially hotspots are real. So this is sort of, let's go over that. We can predict hotspots. And then comes the question, what helps in order to do that? And clearly, sort of the big improvement is from putting it all together. Just evolution does something, just sequence does something, hydrophobic moment and the biophysical features that you talk about is a small contribution. But as you see, putting them all together really is the thing. So none of them in isolation, the biophysical features alone didn't work, but the biophysical features together with something else, and this is sort of the final big step for some cases for which we had the structure, we can even do better. And I believe this is where we'll leave it today. If there's any question about this slide, is there any question? Then I would bring it up Tuesday. If not, then we leave it here.